Well, good afternoon and or good evening, wherever you are. One of the great things about this event is that people from all over the country in different time zones, sometimes different parts of the world. Welcome back to Basic Science Lights the Way. I'm delighted, as always, to see you again for another time of thought-provoking conversation. Tonight, we're going to hear from three biological scientists about their research as it pertains to the great events in evolution. And evolution is a thread connecting all of the life sciences and some physical sciences, many physical sciences as well. And Berkeley is the quilt where those threads intertwine. And I'd just like to add something here that is very important, I think, in discussions about what are the general principles that life sciences or biology have given to all of the sciences. I would say that we owe to Charles Darwin and his colleagues of the 19th century the idea of evolution. And the concept of evolution has woven its way into not only geology and earth studies, but physics and astronomy. And I think it's a basic gift that biology has given to all of the core sciences. And the Berkeley Biological Sciences Division, sponsoring this event, studies everything that breathes, blinks, swims, grows, lives, and dies, from base pairs to the entire ecosystem. Here, evolution isn't just something that happened, it's happening right now at many different levels. Our moderator tonight is Mary Firestone. Mary's a professor of the Graduate School in Environmental Science, Policy and Management, and Plant and Microbial Biology. Mary has a research focus on ecology of microbes in the soil and their contribution to carbon and nitrogen cycle in terrestrial systems, which of course involves, it focuses and impacts evolution. In particular, her lab is interested in understanding the complex interaction between roots and soil microbes and how the crosstalk between roots and soil microorganisms contribute to the health of plants and soils. She also examines the biophysical mechanisms of bacterial interaction with the soil environment. How do the physical characteristics of soil, solid dry soil matrix, affect the growth and activity of microbes? Mary is one of our distinguished faculty on campus, is a member of the National Academy of Sciences. And this past year, she received the European Geosciences Union Duchafor, I probably didn't pronounce that correctly, Duchafor Medal for Soil Science, as well as the American Society for Microbiology Environmental Research Award. Once we get underway, I encourage our guests to post their questions for our speakers in the chat box. While we won't get all of the questions in 1 hour, we'll answer as many as we can, both live and in the chat. And with that, I'm going to hand things over to Mary. Mary, the show's yours now.
Thank you, Mike. I'm pleased and excited to host tonight's event. I think everyone will be impressed by the dedication, creativity, and most importantly, by the curiosity that drives these scientists. Our focus this evening is on great events in evolution. Biologists and naturalists at UC Berkeley study how life on Earth, from microbes to humans, has evolved and adapted through time. We do so from multiple perspectives: genes and molecules, physiology and morphology, individual and populations, ecology and ecosystems. How have humans evolved and adapted to diverse or extreme environments? How have other species and habitats changed over time? And what might that mean for life in the future? It is a holistic approach to doing science and to understanding our world. At the end of each talk, we will try to answer all of the questions for the audience, so please add your questions to the chat. Our first speaker is Professor Charles Marshall. Charles is a professor and Philip Sandford Boone Chair in Paleontology, Director of the University of California Museum of Paleontology, and Chair of the Berkeley Natural History Museum. He is a paleontologist and deep-time evolutionary biologist, broadly interested in how life has evolved on Earth and the processes responsible for changing it and shaping it. Research in the Marshall Lab takes advantage of data from the fossil record, molecular phylogenies, genomics, developmental biology, computer simulation, and functional studies. His research also has a strong epistemological component, often involving the development of new tools or ways of integrating disparate data. Charles, what would you like to share today?
Thank you so much, Mary and Mike, and welcome everybody. Let me just share my screen. So I am fascinated by the history of life on Earth, by life's biodiversity, but I'm also fascinated by the challenge of trying to find meaningful generalities that account for this history. And so what I'd like to do in my brief time with you tonight is outline, first of all, Darwin's theory of natural selection, then give you a brief account of the history of life on planet Earth. And through that process, I want to conclude with an emerging generality that I think transcends the typical view of Darwinian evolution, at least at macroevolutionary scales. So let's talk of Darwin's and Wallace's argument. Quite simply, and this is Darwin's words, as many more individuals of each species are born than can possibly survive, too many baby mice or rabbits, and as consequently there is a frequent recurring struggle for existence, not all of them will make it to adulthood, it follows that any being, any juvenile, if it vary however slightly in any manner profitable to itself under the complex and sometimes varying conditions of life, will have a better chance of surviving and thus be naturally selected. The smarter, the faster, those that learn to cooperate. From the strong principle of inheritance, like begets like, any selected variety will tend to propagate its new and modified form. And thus, Darwin concluded, under the sometimes changing conditions of life, we expect descent with modification, that is evolution. Now, the standard interpretation of the syllogism is that there is no reason to assume that evolution comes with some objective notion of improvement. Or to say it a different way, the only thing that is constantly improving is the adaptedness of individuals to their given environment. Because the environment changes, this progress always remains relative. That is, there's no directionality and no purpose, even though we know organisms individually are highly purposeful. So what I want to do now is give you a very brief account of the history of life on planet Earth, and then make an argument there is, in fact, a kind of directionality in the process. So 4.5 billion years ago, the Earth and the solar system sun form. About 30 to 50 million years later, a collision with a small planet called Theia created the moon from the debris, and also the seasons by knocking the axis of the Earth from vertical to its roughly 23-degree orientation. Somewhere between 4.4 and 3.5 billion years ago, life originated, quite possibly within the porous deposits at alkaline hydrothermal vents on the ocean floor. The age is poorly constrained because we have virtually no rock from this time period. There are data to suggest that cells actually evolved twice, and thus first life inside these hydrothermal vents was acellular. Now, important in this talk is the fact that there are two types of cells. There are prokaryotes, relatively simple cells such as E. coli or Salmonella, and the cells of our bodies, eukaryotic cells that have between 1,000 and 100,000 times more DNA and between 1,000 and 1,000,000 times bigger volume. 3.5 billion years ago, we have the oldest fossil evidence of life, fossilized biofilms that are called stromatolites, sometimes associated with microfossils. So for the first billion years of life on Earth consisted of these prokaryotic cells, and they were limited feeding by diffusion. They're simply too small to hunt. And so here are some modern stromatolites from Western Australia. And if one is to cut them, shown here, they have a layered structure that is often seen in the fossil record. About 2.5 billion years ago, oxygen begins to build up in the atmosphere and the ocean for the first time after the rusting of the oceans, but only at 0.1 to 1% current levels. Initially, the ocean was filled with Fe2+, which was absorbed by the ocean, which is what I mean by rusting. Initially a poison, oxygen enabled a much more powerful metabolism, which we now thrive on. This great rusting of the ocean has led to the world's great iron ore deposits, as well as uranium, etc. For example, indicated by this iron ore mine in Western Australia. At about this time, roughly 2.5 billion years ago, the first eukaryotic cell emerged, a consortium of two different types of prokaryote, shown here, and through a process that is not yet fully understood, leading to eukaryotes themselves. The earliest fossil eukaryotes date to about 2.1 billion years ago, indicated here. And then we jump to just 700 million years ago, the first animals. It's currently believed that these are sponges, but as you'll hear from our second speaker, Dan Rokhsar, they may well be a different type of organism. Sponges have feeding cells that look an awful lot like single-celled choanoflagellates, and so we think the sponges emerged from this particular group of single-celled eukaryotes. 580 million years ago, more complex animals evolved, but with simple ecologies, but complex genomes. 540 million years ago saw the Cambrian explosion, the emergence of complex animal phyla, approximately 30 of them. And this beast here is called Opabinia, with 5 eyes, spectacularly preserved in the soft-bodied fauna of the Burgess Shale in Canada. And here is Opabinia here in the preservation here in the Burgess Shale. About 500 million years ago, algae colonized land. This is a cartoon of the first forests. 430 million years ago, insects moved onto land. About 365 million years ago, vertebrates then follow those insects onto land. The first terrestrial vertebrates, our ancestors, evolved from fish like these that have muscle and bone in their fins, so-called lobe-fin fish. About 225 million years ago, the first mammals and dinosaurs appeared after the end Permian mass extinction. All were small, especially the mammals. And I have here a paper clip for scale for the earliest fossil mammals that we know. And then 225 to 66 million years ago, in the so-called Mesozoic era, dinosaurs dominated the land. And then at 66 million years ago, a meteorite the size of Mount Everest hit the Earth, ending the reign of dinosaurs and the Cretaceous period, ushering in the Cenozoic era, where mammals came to dominate the land. About 2.5 million years ago, our own genus, Homo, emerged. And roughly 300,000 years ago, our own species, Homo sapiens, emerged. The fossil record is highly informative, but there are amazing revolutions happening in ancient DNA. And our third speaker tonight is going to talk about ancient DNA as it informs the evolutions of humans. This is Rasmus Nielsen, recently elected to the National Academy of Sciences for his work, especially the computational analysis of these DNA sequences. So what then, in conclusion, does the fossil record have to tell us about the history of life? It reveals a long-term directionality to evolution, the increased power of the most powerful species, and through that, increased power of entire ecosystems. The notion that selection favors the powerful was first posited in 1925, but as a general principle, it hasn't yet gained proper recognition. Eukaryotes are more powerful than prokaryotes. Warm bloods are more powerful than cold bloods. Flowering plants are more powerful than pine trees. And we are vastly more powerful than any other species. And this is beautifully explained and illustrated in an upcoming book by Gary Vermeer of UC Davis. So look for that if you want an enjoyable read. And so finally then, as we look at the trajectory of the history of life from the formation of the solar system to the early biomolecules to the evolution and origin of life, here are stromatolites sitting here, marine organisms, the first vertebrates to gain land, the dinosaurs, and into the mammals, and then humans and our capacity to apprehend that history, there is a long-term trend of increasing powerfulness, which seems to be the general principle under which life has emerged. And with that, I am open to questions. Thank you.
There are several questions that have come in and are very interesting. What is the primary evidence for Thea's existence and its collision with Earth?
So the primary evidence is that the Earth has a crust density of about 2 tons per cubic meter, and then a mantle, and then a very, very heavy iron nickel core. The moon should have a relatively same composition if it was formed in the same way, but it's basically missing the iron nickel core. And so the idea then is that we were hit after our core formed by a third body that blew sufficient material off that then condensed into the moon, accounting for the lack of a core, its relatively low mass given its size. The other piece of evidence is that when moon rocks were returned from the Apollo missions from the moon, it's almost like the moon's detailed isotopic composition looks like a twin of the Earth, suggesting that in fact that material was blown off the surface of the Earth to recondense as the moon. So those are some of the primary pieces of evidence for a collision creating the moon and the tilt of the Earth's axis of rotation.
Thank you. How did oxygen shift from being a poison to a power source?
So it started off being a poison, as it is today for things that live in anaerobic environments. But life is enormously creative. And we learned to harness the oxidative power of oxygen to produce what's called oxidative phosphorylation. So organisms get very, very good at basically nullifying the effect and then using the fact that it burns, internalizing it. I don't think I'll go into any further detail than that. Mary knows this answer better than I do.
I'll keep my mouth shut. Why do you propose that life likely originated in an alkaline seafloor setting?
Complicated. To me, life needs containment, it needs energy flow, and it needs material flow, and it needs those in prodigious quantities. And these alkaline hybrid thermal vents have those, and they specifically have hydrogen and CO2, which is the foundation of the earliest metabolic pathway, the acetyl-CoA pathway. So I think life originated inside these containments, and then cellularization happens twice. The cell membranes of archaea and bacteria are built differently. The locomotory devices are completely different, like a bird's wing versus a fly wing. And this also accounts for the fact that those are completely different if cellularization and locomotion occurred independently twice from some ancestral home.
Thank you. What does powerful mean for fitness? We need a definition, says Jeffrey Mark.
So power is basically just the work, energy per unit time. And so I need to acquire energy to conduct the processes of life, reproduction, gaining food, excretion, protecting myself. The more energy I can acquire per unit time, the more work, the more powerful I am, the more successful, the more offspring I'm going to leave behind. And so I would use power in that simple way. You can measure it by reproductive success, which is measured by biomass, which is a concentration of energy per unit time. So those are the units that I would use formally. And I would like to write some equations down, but I don't think that will work for this setting.
Okay, I think we have time for one or two more questions. What factors precipitated the explosion of diversity? This is a question from Sheldon Green.
I think the explosion of diversity fundamentally comes from the fact that there are trade-offs. It's very hard to be a master of all trades. And so for fish, for example, you can have very powerful jaws, but they're slow, or you can have very fast jaws, but they're not so powerful. And so you get two fundamental types of feeding in fish. And so when you look at any set of organisms, given the multiple functions they have to perform in order to be reproductively successful, there are a huge number of combinations of trade-offs. And it's that huge number of combinations of trade-offs that's responsible, I think, for the enormous diversity of organisms that we see today.
Okay, then one more question from Edwin Ching. How did the evolution of viruses occur over these timescales?
Viruses. Tricky, tricky, tricky. They are so simple that we don't see direct evidence of their origins in their DNA sequences. They evolve so fast and they're so tiny. So it's an open question as to whether or not they're relatively recent arrivals on the scene, where they're super reduced sort of parasitic forms. My suspicion is they've been there almost from the beginning, but we don't really have a good understanding of their origins or how many times they've originated. So that's a question for the future.
Okay. Thank you, Charles. That was
You're welcome.
a fascinating presentation. Our next speaker is Daniel Rokhsar. Daniel is the Marthella Folkett Brown Chair of Biological Sciences and Professor of Genetics, Genomics, Evolution, and Development in the Department of Molecular and Cell Biology. He was originally trained as a physicist and is also a faculty member in the Department of Physics at Cal. His research is focused on understanding the origin, evolution, and diversity of animals by combining computational genomic analysis with comparative developmental biology. He is especially interested in the evolution of novelty. Daniel, please tell us about your work on the deep time evolution of animals.
Thanks, Mary. So my obsession is to understand some of the events that Charles described, and to try to bring to bear a comparative approach to figuring out what happened, say, 500 million years ago, which is not as long as some of the events that Charles discussed, but still a pretty long time ago. So our starting point is, as Charles mentioned, is this idea that life is related by a phylogenetic tree, shown here in the upper left by the only diagram in Darwin's book. I love the thought. It's "I think," and then his thought is a picture. And this picture illuminates a lot of how diversity appeared on our planet. And here's a little vignette that gives you some idea of the power of this idea: tree thinking and comparative analysis across mammals. So here's a bunch of pictures of different mammalian species. I like the one in the middle as my favorite. Those are baby hedgehogs. And you can see that this is a tree. It's a little bit more rectangular than the one that Darwin showed, but it shows you the relationship among many of the different major mammalian lineages. You're here on the far right-hand side. And so if I look at these, I see enormous amounts of diversity, right? Animals that fly, mammals that fly, mammals that swim. But I know that they all came from a common ancestor of all living placental mammals. These are all mammals that give birth to their young live. And that liveness, the live birth aspect, probably therefore arose in the ancestor and is then preserved in all the remaining lineages. So by looking at this comparative approach and figuring out what's shared among a diverse group of animals, I can say something about the ancestor that lived, as Charles mentioned, maybe 150 million years ago. I've lost my Zoom. If I want to look more deeply in evolution, I have to look at a much broader diversity of animals. And so shown here is a cartoon of the relationship among different kinds of animals. The ones that are shown in these green, blue, and orange boxes, those are the animals that most of us think of when we think of animals. These are animals that have a head and a tail, a central nervous system, some kind of a brain, and generally move around. And to a first approximation, most of these animals are what you would think of as worms. But among those worms, there's a lot of diversity. So I've shown you some pictures up here on the top just for orientation. Arthropods include flies, mollusks include octopus, echinoderms include sea stars, and you are here in the vertebrates. And many of these animals, these pictures that I show you, are organisms whose genomes we've sequenced in my lab here at Berkeley to try to understand the diversification of the DNA that these animals contain, and in that way try to triangulate our way back to this common ancestor. So by that principle that I just mentioned in the previous slide, these animals all have a head and a tail in some fashion or another. Therefore, it's likely that their common ancestor who lived before the Cambrian explosion probably also had a head and a tail. And so in that way, I can say something about this common ancestor, even though the fossil record is really hard to interpret because there were relatively few, all the animals there were soft-bodied, and so there's relatively few fossils that I can compare to. If I want to go back even further in time to the first animals, which is represented by the 700 million year branch A, now I have to bring in other kinds of organisms, ctenophores, cnidarians, placozoans, porphyrians, or sponges. These are other kinds of animals that are very different from the animals that you normally think of, and I'll talk more about them in a further slide. And then if I'm continuing to think about the phylogenetic tree, as I reach further back in time, the next branches are going to be not animals at all, but single-celled organisms, organisms that don't have the complexity in the organization of animals. So if I want to look at these very deep moments in time, I have to look very broadly across a huge swath of organisms and be able to figure out what it is that they share. Then I can use that to triangulate back to what their common ancestor was like. That's the principle. So what I'd like to understand are two big events in evolution. First, how did animals evolve from non-animals? There was a time in the oceans where there was nothing that we would recognize as an animal. And then millions of years later, there were animals. How did that transition happen? And then similarly, but a little bit harder to think about, is how did vertebrates, animals with bones and backbones like us, evolve from invertebrates that are squishier? And so I want to figure out these events, both of which occurred about 500 or more million years ago. I need to have something where I can compare broadly across animal diversity and figure out what's shared. And then by knowing what's shared, I can start to figure out what the ancestor was like, and also what how these features have changed. And so a general feature of all animals, of all life, in fact, is that it contains DNA, in some cases RNA for viruses, sequences, and I can use and compare those DNA sequences as a uniform feature that I can study across a broad range of organisms. But DNA sequence changes rapidly. And if I want to look 500 million years into the past, I can't use the DNA sequence itself. That's too labile, too quickly changing. But I know that genomes are also organized into chromosomes. And so we have the idea that maybe it's the chromosomes that are changing slowly enough that we can use them. Now, I don't have time to go into a lot of details, but humans famously have 23 pairs of chromosomes. You have pair because you get one set from your mother and one set from your father. But this number 23 is specific to humans. Other species have different numbers. Sorry, my Zoom is flicking out. So if I look at the chromosomes of human, this is a cartoon of the 23 human chromosomes with different colors, and I compare them, say, to mice. If I do this comparison, I can see that I can ask, where are the genes that are on chromosome 1 of humans on the mouse genome? And it turns out they're split across 3 different chromosomes. And in general, you can compare this color coding here in mice to what it is in human. It's as if the chromosomes have been shattered into 100 pieces and joined back together in a different way to yield the 20 chromosomes of mice. So this suggests that chromosomes are not a really good thing to look at because they're changing too rapidly. This is only 100 million years of evolution. I want to go to 500 million years. But over the last decade or so, we've been looking at other kinds of animals, mollusks like scallop, Amphioxus, which is a kind of a marine worm, sponges, which are one of those early branches I mentioned, jellyfish, hydra is another species. And what we're finding is that, somewhat remarkably, chromosomes are relatively stable, very stable, once you look outside of mammals. And so if I follow each of these threads as an individual gene, and so I follow, say, the genes that are on this purple chromosome in scallop, they're also on the same chromosome in Amphioxus, and they're in the same chromosome in sponge. So these 2 organisms, sponge and scallop, shared a common ancestor 500-600 million years ago. And this purple unit has remained intact over time. Using that, we can now attack this problem of what were the earliest animals. And here are the candidates. I've got sponge on the left, doesn't move around, filter feeds in the ocean. I've got comb jellies, which swim around, placozoans, which crawl around, cnidarians, like jellyfish. And then here for biologists, I've used Rube Goldberg, class of 1904, sort of a patron saint of evolution, who figured out complicated ways to do things that work, and that's what matters. And each of these different animals must have had emerged from this common ancestral node, which I mark here at the question mark. But until recently, there's been a lot of controversy about how these branches occurred. I'm happy to talk more about it if there are questions, but we've been able to show by looking at the chromosomes that these animals here, sponges, placozoans, cnidarians, and us, bilaterians, all share certain chromosomal features that are not shared by jellyfish, by comb jellies. That implies that the comb jellies must have been the first branch, contrary to the conventional wisdom, which had sponges branching first. And the importance of this relates to a key characteristic of most animals, or all animals except for sponges, which is that how nervous systems are distributed across the tree of life. So comb jellies have nervous systems, sponges don't. They just sit there. They don't have the need to integrate that kind of information. Placozoans are not believed to have a nervous system, but cnidarians and bilaterians do. So according to this new picture, we have nervous systems here and nervous systems here on the right. That either means that the ancestor of all living animals had a nervous system that was lost on the sponge lineage, and probably trichoplax, or more controversially, that the nervous system arose twice. So this is my last slide here. This is a picture of a museum in Paris that I visited before the pandemic, and a great quote from Ted Chiang. I'll let you just read it. If I compare all these animals, just looking at them, I can get a sense of what their last common ancestor looked like. These are all vertebrates, but using genomes, we can look even further back in time. Thanks.
Thank you, Daniel, for a wonderful talk. There are a number of questions for you, some still coming in. What kind of organisms are placozoans?
Sure. So placozoans are very small animals that are known from mangrove forests and other marine environments. And to find them, you put a glass slide out into the water, you come back a few weeks later, and you'll find these little microscopic creatures crawling around. Unlike all other animals, they're 2 cell layers thick, and they crawl around, and basically it's as if you imagined an animal whose gut was sticking out underneath it. And so it doesn't have a mouth, it doesn't have a way for waste products to go out the other end, it doesn't have a gut at all, but it crawls around. Trichoplax means flat and hairy, and that's the simple characteristic of it. Their genomes are unusually complicated, and it's unclear exactly how these animals reproduce, and so maybe there's another form out there that we haven't yet discovered.
Okay, thank you. A good question from Morgan Schwartz. What are the techniques for dating the evolution of genes?
Hey, Morgan, that's a great question. So when you look at gene sequences, if you're looking over relatively short times, so say you're looking, as Rasmus is going to tell us, short from on my scale, we're looking at changes that have happened over hundreds of thousands or a million years. We often know from the fossil record when different lineages have diverged. So if I compare, say, the sequence of a chimpanzee and a human, and I see how much change has happened, I can go to the fossil record and I have some rough idea of when that happened, 7 or 8 million years ago, some number like that. So that gives me a kind of a clock to calibrate the change. So if I see a certain amount of change, I can translate that into time. The further back you go in time, the harder it is to do that, because you get change layered upon change, layered upon change, and you have to make mathematical corrections to adjust for that. So I think it's a complex analytical process to do that gets harder and harder the farther back in time you go.
Okay. Daniel, can you briefly describe your research about the emergence of vertebrates?
Yeah, I'm not sure I can do it briefly.
In 3 sentences or less.
Yeah, so there was a time when there were no vertebrates, and then there are vertebrates, and that there's a gap, what I would consider a sort of a weak region of the fossil record, where it's really hard to figure out how that complexity arose. And so we've been looking, using the same principle, at animals as diverse as hagfish and lamprey and a bunch of other fish that you may recognize. And what we're finding is that a key event is the genomes doubling and doubling again. There's an enormous amount of duplication, something that we know of a lot of in plants, but it really seems to be a key event in the origin of vertebrates.
Okay. Edward Ching would like to know how useful are non-coding regions for your analyses?
Yeah, so that's another great question, Edwin. As you know, the non-coding regions of the genome are responsible for controlling when the genes get turned on and when they get turned off. So a lot of the complexity of animals arises from the orchestrated choreography of genes, depending on these non-coding regions. Unfortunately, for my interests, these non-coding regions evolve very rapidly. Their sequences change very, very fast. And so we've looked at this in various plant sequences and some animals and tried to understand that. But if you want to go 300, 400, 500 million years, those sequences become very difficult to recognize and hard to work with. So unfortunately, that seems to be a little bit out of our grasp.
Okay, this is probably the final question we're going to have time for. What proportion of traits evolve slowly enough to enable comparative genetic analysis, say, versus fast evolving adaptations?
Yeah, that's a really good question. And we've been focused on trying to find traits that evolve slowly. Like, so for example, the animals that have a head and a tail almost always have a mouth, a tube, and then a hole at the other end. And that's how food passes through. So that's a characteristic that once it evolved has very rarely been lost. So in a sense, that's evolving slowly. I think it's really hard to put a number on it. We're always on the lookout for features that we can use to try to do this sort of analysis I described, which then gives you a framework for thinking about how evolutionary change has happened in all the other characters.
Okay. Thank you, Daniel. And
Thank you.
thank you also to our audience for the interesting questions. Our final speaker is Rasmus Nielsen. Rasmus is a professor in the Department of Integrative Biology. His research focuses on statistical and computational aspects of evolutionary theory and genetics. One of the central problems for studies is the molecular basis of evolutionary adaptation. What happens at the molecular levels as one species is transformed into another over evolutionary time? To address this question, the Nielsen Lab works on the development and application of statistical and computational methods for analyzing genomic data in the context of evolutionary genetics, population genetics, and medical genetics. Over to you, Rasmus.
Thanks so much, Mary, for the introduction. Let me just share my screen. So I'm going to start by revisiting this tree, Darwin's tree, that Dan Rokhsar told you about. We are all, as evolutionary biologists, we've all been taught to think of evolution as a tree-like process of species that diverge from each other. Researchers that work on bacteria, and know that that story is not as simple as that. Bacteria often evolve in a non-tree-like fashion. And what we've learned recently is that's actually true much more generally. For example, us as humans, we have not evolved in a tree-like fashion. And I will talk to you today about how we got to that conclusion. So for me, it started, hold on a second, I just need to change my screen. It started in a collaboration with Svante Pääbo, who sequenced the Neanderthal genome. And as part of the collaboration with him, we analyzed the DNA data and tried to identify regions of the genome that was shared by Neanderthals and humans. And what we were able to show is that humans that live outside of Africa or trace their ancestry to outside Africa, today they have 1 to 2% of their DNA from Neanderthals. So the conclusion is that humans and Neanderthals, they interbred actually with each other. And if we look at the human evolutionary tree, we can now draw a branch between the Neanderthals and the ancestors of people with ancestry outside of Africa. So that's what we call introgression, the transfer of genetic material between species through interbreeding. Now, one of the things that we study is human adaptation. We study many different population groups and try to understand how they genetically have adapted to the environments they live in. And as we did that, we time after time saw something similar having happened for humans in different environments, that the DNA that they got from other hominin species had helped them to adapt to these different environments. And I'm going to tell you today about one example of that. That's studying our studies of Tibetan altitude adaptation. So on the Tibetan plateau, that's in an altitude about 4 kilometers high, the amount of oxygen you get when you breathe is only about 60% of what it is at sea level. So that means that when you go up there, you will be exposed to acute hypoxia, low oxygen levels. Part of your response to that is to produce more red blood cells. But the Tibetans are very special in this sense. They have a completely different regulation of red blood cells and red blood cell production. And what we're able to show is that the particular gene that's called EPAS1 has changed in the Tibetans to allow them to regulate the red blood cell production differently. This gene EPAS1 is what's called a transcription factor. It tells other genes what they should be doing, and particularly it regulates the production of red blood cells when there's low oxygen. So this is great. We found an example of a gene that has been evolving recently in humans. And what we do then is we're trying to model that mathematically to try to answer questions about when selection started acting on the gene. Is the selection only acting in Tibet? And how strongly is the selection acting, and so on? Now, the problem we ran into was that none of the models that we have, the mathematical models, could actually answer these questions. Somehow there was some pattern in the data that we just couldn't explain with any of these models. And I'm going to explain to you what the pattern was that we saw that we couldn't explain. So this figure illustrates some properties of the data. We are comparing the Han Chinese from China to Tibetans. And these lines here are supposed to be DNA sequences. And what we found was that there was a long segment around this gene called EPAS1, where the Tibetans were very, very different from the Han Chinese individuals. And none of the models, the mathematical models we have of how evolution works, could really explain that pattern. So we thought maybe this could be from Neanderthals. But when we compared to Neanderthals, there was actually no match. But then it turned out that as Svante Pääbo worked more on sequencing Neanderthals, he actually found DNA from another hominin species from the Denisovan cave in south-central Siberia. We now call them Denisovans. We mostly know them from a few remains, like this little piece of a finger bone and a few teeth, and then from their genome. And so what we could do is we could compare these Denisovans to the DNA data that we had. And when we did that, there was a near perfect match between the Tibetans and these Denisovans. So what we could show was that humans in Tibet that had adapted to the low oxygen environment in Tibet, one of the main reasons why they could do that was because that gene had been transferred from these Denisovans into the Tibetans by interbreeding. That's an example of what we call adaptive integration. So that is adaptive, meaning it provides an advantage to the individuals getting it, and integration, that's this transfer genes between species. Since then, we looked at many other examples, and other groups have done so as well, where we've found again and again, as humans entered particularly extreme environments, one of the reasons why they could adapt to those environments was that they could pick up genes from other hominins that were already there, like Neanderthals and Denisovans. And by doing so, they could fast adapt to these environments. So this process of adaptive integration seems to be really important in human evolution, and is one of the reasons why we've been able to spread around the world so fast. Now, it's not just humans doing that. In other studies we're doing on other animals, we tend to see that again and again. It's one of the big lessons that we learned from genomic sequencing, is that units that we thought were very separate species, that actually interchange genes all the time. For example, here, the Bos genus, so that is cattle and their relatives, they have exchanged genes between species that we consider very different from each other. For example, the yak and the cow, common cattle, domesticated cattle, where there's been exchange actually of also EPAS1. So that one of the reasons why cattle in Tibet can function better in Tibet is that they also got EPAS1 from a species that was adapted there from the yak. But remarkably, the same thing also happened for dog. Tibetan dog also got this gene EPAS1 from the Tibetan wolf. So the same gene has been transferred between species many times to allow new species to adapt to the low oxygen environment in Tibet. So where we're at today is that we have to revise this tree that Darwin wrote. We have to add lineages here and there and everywhere. So we no longer has a tree. What we really have is a network. So we have to revise the way we look at evolution as not being tree-like, but much more network-like. Thank you for listening to me today.
Thank you, Rasmus. Several questions for you. Do we know if the Denisovans interbred with other groups of humans besides Tibetans?
Yeah, actually, you see most Tibetan or most Denisovan DNA in people in Papua New Guinea and in Australia, indigenous people in those regions. They actually have as much as 3 to 5% of the DNA from Denisovans, whereas in East Asia, they have very little DNA from Denisovans, but very important piece of DNA, for example, for the Tibetans. But there are other places in Southeast Asia and Australia where they have much more Denisovan DNA than what we see in East Asia. And if we look at Europe or Africa or the Americas, we don't really see any Denisovan DNA at all.
Okay, thank you. Interesting question. What features of the modern human genome have come from introgression with Neanderthals?
Well, several different important genes have been transferred from Neanderthals to humans, and one of the several of them have to do with immunity and defense. And in fact, one of the genes that is causing differential response to SARS-CoV-2, the virus that causes COVID-19, that was transferred from Neanderthals into humans. So what we think happened is that when humans and Neanderthals met each other about 50, 60,000 years ago, first in the Middle East, with that first handshake they gave each other, they transferred a lot of pathogens. And we see that it's often when separate populations come together, that often pandemics spread then because it transferred pathogens to each other. So that's probably because of that, it was advantageous to, if you could pick up the genes from Neanderthals that allowed them to have some immunity to the Neanderthal pathogens, that would be beneficial for the humans that did that. And so one of the processes that might have been driving this integration of DNA from Neanderthals into humans might actually have been the selective pressure to be able to better protect yourself against the Neanderthal pathogens.
Okay, thank you. A very interesting question from Morgan Schwartz. How are you planning on, or how are you using AI, or how could we use AI to interpret DNA patterns?
Well, we are using machine learning methods in many different ways to understand patterns in genomes. For example, if we're going to find these segments of Neanderthal DNA that is in the human genome, how do we best do that? And that's one of the approaches where machine learning methods have been really powerful in going in and picking up what are the pieces of DNA that is actually from Neanderthals in the human genomes. But we, of course, it's being used many other ways. It's being used for predicting disease or disease susceptibility, for predicting interactions between genetics and the environment in disease susceptibility and so on. So AI, or at least machine learning methods, are being used constantly for interpretation of human genomes.
Okay, thank you, Rasmus. Very interesting. And thank you also to Charles and Daniel for sharing your research with us. Evolution is truly a fascinating topic. We have a few more questions for the audience, for the speakers. An interesting one from Rosette Dawson. For Charles, does power need to be balanced with altruism for a community's ability to survive? Are humans going to power themselves out of existence?
Yeah, that's a very good question. I think we're seeing with humans what's happened in evolution over millions, tens, hundreds of millions of years, sort of almost within our lifetimes in the last century or so. So there's always a balance. Whenever you see anything stable anywhere in the universe, it means there are opposing forces. Stars, gravity pulling in, radiation pressure going out. And so there's a trade-off between acquiring power and efficiency. And there's a trade-off constantly with the availability of resources that enable you to generate power in the first place. If you're a predator, there aren't any prey items, you're in trouble. And so, yeah, I don't think we're going to power ourselves out of existence, but we're clearly running into the limitations of power. And we don't yet know how to talk about that particularly sensibly as we talk about how to generate more power and more power and more power, rather than talking about how do we actually live within a certain amount of power. It's a very important question.
Thank you. A question for Rasmus from Jeffrey Marquis. What are the chromosomal constraints on interbreeding between species?
Yeah, so if the chromosomes, and Dan Rokhsar could actually talk probably even better than me about this. If the chromosomes don't match, what will happen is that as the cell divide and produce gametes, so sperm cells and egg cells, it's very likely that you will get the wrong complement. That process will go wrong. We'll get the wrong number of chromosome in these gametes. So essentially, you will produce cells that are not viable. So typically, for species to be able to interbreed and a viable offspring, you need chromosome complements that matches each other. Otherwise, the offspring will be infertile because this process of producing these gametes will go wrong.
Let's take some general questions to all three of the speakers. Do you have a favorite seminal event in the history of life on Earth?
That one has to be for Charles.
Yeah, origin of life, origin of eukaryotic cells, origin of multicellularity, origin of humans. No, it's the continuity and just this constant expansion of capacity is really what drives me, fascinates me.
Okay, I would like actually each of you to answer this briefly. What inspired you to study the science of evolution?
Well, I can go first. I came to biology from having studied physics, and in physics we're used to having general principles, and so we understand a new situation in terms of that general principle. And when encountering biology, everything just seemed so diverse. It was unclear what was tying everything together. But as Dean Botchan said at the beginning, evolution gives you this framework for thinking about all aspects of biology. And so it's the unifying principle. And as a physicist, I'm drawn to that.
Yeah, I was one of these kids always running around catching frogs and lizards out in my backyard and spending much of my childhood and my youth out in nature and being fascinated by it. And of course, you want to understand what's around you. And if you're not very deeply religious and want to understand the diversity of life, you have to become an evolutionary biologist. So I didn't see any other choice for me than becoming one of those.
And for me, it was dinosaurs. The fact that the world has changed unimaginably from today, those frogs, to things that we would have no idea existed if it wasn't for the fossil record. And so for me, it was the transformation of life, dinosaurs back when I was 4 or 5, that got me hooked.
Thank you so much. I wish we could continue this conversation. It's fascinating, but in fact, we don't have geologic time here this evening. So to close, I'm going to hand the floor back to Dean Michael Botchan. Mike?
Well, yeah, Mary, I wish we had not geological time to discuss this, but at least another couple of hours. I think it could go on a lot longer. But we all have to go on with our lives, and looking forward to great excitement in the future on this general topic. And I want to thank you, Mary, for the job you've done as moderator, and Charles, Daniel, and Rasmus, your unique approaches to this very deep problem in life sciences is much appreciated. And I just want to tell the audience that I actually put something into the chat that I wanted Dan Rokhsar to talk about, but he didn't answer it. And maybe
I sent you a private answer.
Okay, there you go. All right.
I can tell everybody if you want.
Well, maybe no. Let's end a minute or two early. So, and I especially want to thank our audience for joining us this evening. I encourage you to reach out if you'd like to learn more about anything that we've covered. We'll be sharing additional resources with you by email about today's topic and speakers, and a video of today's session will be available at our Basic Science Lights the Way website in approximately a week from now. And I'm going to be going to New York City soon to visit with my grandson, who's now turning 12, and I'm gonna actually have him watch the video when we can. And so I think it's, it's something that is a topic that should influence all human beings who have the power and eat enough food to focus on it. Tonight's the final event for this season's Basic Science Lights the Way series. And it's, I think, nice that we've ended with such an important topic that is so general. We began by hearing some tales from new faculty members a while ago, and we learned about interesting ways that mathematics are being applied to different fields. And for me, there are basic questions there about why equations can actually tell us about reality, what's in that. And we heard from, it doesn't have to be that way. It could have been that everything is chaotic and math doesn't really help us at all, but it does. And we talked in marvelous ways about how the body can repair itself and regenerate itself. And I found these events and our speakers to be remarkable. And I hope that you have too. You have also found it exciting, and we encourage you to keep joining us. And until we see you again next fall, stay curious and fiat lux, and go Bears.